Hey guys, good morning. Hope we're doing well. We are going to continue with our mitosis PowerPoint unit. We're going to talk about cytokinesis today and a couple of other key points. Before we do that, let's quickly review some stuff that we did yesterday. So yesterday we spoke about mitosis. We spoke about interphase. We spoke about prophase, metaphase, anaphase, and telophase. Please make sure you know what each of those are, what happens in each of those phases, and how to identify them if you needed to. We are going to do a little bit of a review, see if you remember some stuff. So as I go through this, feel free to pause the video to see if you get the answers correctly. Before we do that, though, let's do just a quick recap. Normally, if we were in class, this would be blocked out, and I would have some of you try and tell me what the names of these phases were. We cannot do that, unfortunately, so we're just going to go through it. But we do have a couple of our do-nows after this, so it kind of feels like we're back in school. All right, so this is metaphase. Remember, metaphase, the way to identify metaphase is everything is lined up in the middle. Notice that everything is lined up in the middle. This is a really good picture to look at, and this is a good point because it doesn't necessarily need to be horizontal. It can be vertical as well, like you see here. Just if it's lined up in the middle, then it is metaphase, right? And you see in this one, this part is also metaphase. This one happens to be horizontal. This one is vertical. They are both metaphase because they're both lined up directly in the middle of the cell. Remember, it doesn't matter which way it's lined up as long as it is lined up in the middle. Here we have anaphase. Remember, anaphase is when the chromosomes and the chromatids start to be pulled away from each other. And you can see in this picture here that they're starting to be pulled away. You have some being pulled up, you have some being pulled down. There's a gap in the middle. That's anaphase. Same thing in this picture. There is a gap, right? And they are being pulled to their respective poles. That's anaphase. Right? Remember, telophase is when there are two distinct cells. A very easy way to identify telophase is again by this pinch point here. Do you remember what the pinch point is? You should remember that it's called the cleavage furrow, right? Telophase. And then again, you have prophase because remember, the centrioles are not on opposite sides of one another. They're just starting to move away. And you can also see that the nucleus is very, there's still a membrane and there's still a nucleolus. So we know we're in prophase. And then in metaphase, again, we are lined up right in the center. So we know that we are in metaphase. Okay. So that is some practice. Again, as part of the work that you had done, you did some practice with this. You will continue to get that. Feel free to Google, identifying mitosis phases. There are definitely a lot of resources you can use. I will try and post some of those as well. But you really do need to know how to identify them. And again, how to describe what is occurring in each one, right? So I mentioned in the other video that, you could get a question on a test, on a quiz, on the Regents where they describe something that's happening in a phase and you have to name the phase. So they can say something like chromatids being pulled apart from one another to opposite ends of the cell. What phase is that, right? And you should be able to say that that is anaphase, right? They could mention a cleavage furrow. You should know that that's telophase. They mention the chromosomes in the middle. That's metaphase. So just know how to describe each of those, and you should be good. All right, so again, what we did yesterday is we went through mitosis. Remember, mitosis is prophase, metaphase, anaphase, telophase, right? PMAT for short, PMAT. Prophase, metaphase, anaphase, telophase, PMAT. Remember, interphase is technically part of mitosis, but it is just the previous phases. Remember, it's G1, S, and G2. It's part of the cell cycle. It's not part of mitosis, right? So it's still part of the overall cycle, but it's not the actual splitting of the cell. It's the growth, the development, the DNA replication phase. So don't forget what interphase is either. That is important to remember. So we went through interphase. We did G1, S, G2. We did mitosis, prophase, metaphase, anaphase, telophase. There's still one part left of the cell cycle, right? And it's called cytokinesis, right? So here we have our cell in anaphase, right? It's at the last, in telophase, I'm sorry. It's at the last phase of mitosis where you have the cleavage furrow. You have two distinct cells, but they aren't split yet, right? You still have one cell technically, right? It's one membrane. That's the real thing to remember. You have one large cell membrane around these two cells. So even though you could see two cells, they have not yet split. This is where cytokinesis comes into play. So at the end of mitosis, two nuclei are formed, and each have an identical set of chromosomes, right? They have replicated, they have moved to opposite ends, you now have two distinct cells. Cytokinesis is the last part of the cell cycle, and this is where the cytoplasm divides, right? It divides. So what we see here is cytokinesis usually occurs at the same time as telophase. However, it is its own phase, so that's why it has its own name and why it's not part of telophase. But it usually will happen concurrently. It could happen a little bit after, depending on the cell, depending on the environment, but usually it doesn't. But it is still important to remember this phase because we are going to speak of it as a separate phase. So cytokinesis is the division of the cytoplasm, right? So what's going to eventually happen is you are going to have the cleavage furrow right here. I just pointed on my screen as if you could see that. You could, okay, so you have the cleavage furrow, right? Right here and right here. You have a cleavage furrow right here, right here. Those furrows are going to continue to move inward. They're going to continue to move inward. They're going to continue to move inward, and it's going to continue to move inward until it just pinches off, and now you have two separate cells. So the furrow is going to keep moving in until the cytoplasm just splits and it divides into two cells. And that's what cytokinesis is. You can have a question asked on a test on the region, what occurs during cytokinesis? Your answer would be the cytoplasm divides so that there can be two separate cells. That's it. The cytoplasm will divide so that there can be two separate cells, right? Because even though there are two cells here, they are still connected to one another during telophase. Cytokinesis is when the cytoplasm divides, the cleavage furrow keeps pinching inward until there are two separate cells. Okay? Now, cytokinesis occurs both in animals and in plants. There is a couple of differences that we're going to talk about. There's really only one main one, but there are a few distinctions. So cytokinesis in animals, remember we spoke about in animal cells, the cell membrane is pinched inward and it creates that cleavage furrow that we see. And that cleavage furrow keeps moving inward, the pinch keeps occurring until two separate cells are created. And again, each cell will contain its own nucleus, its own cytoplasm, and its own organelles. They are exact copies of one another that have now been split, right? So that's cytokinesis in animal cells. We're going to talk about plant cells in a second. What we're going to do now is we're going to go through 3 do now questions, right? We're back in school, so let's pretend like we're in class. Take a look at the question, pause the video, and then see if you can answer it, okay? So the diagram shown represents a microscopic structure observed during the process of cell division. Letter A indicates what? So A is pointing to this part right here, right in the middle. And your answer is centromere. Remember, centromere is the part in the middle that connects these two parts to these two parts. It's right in the middle, right? Centro sounds like center. So it's the center of the chromosome that connects the top part to the bottom part, okay? Let's go through this one. Pause the video, see if you can answer it. Oh, actually, nope, can't answer that one. We didn't do that yet. That is the, this has to do with plants, right? So we're going to come back to that. Should be able to answer this one, though. The diagram shown represents stages of a cellular process. Which is the correct sequence of these stages? All right, pause the video, take a second, figure it out. The answer is C, right? So we have C, B, D, and then A. Now remember, you know that A always has to be last in this sequence, right? Because you see two distinct cells. That means this has to be in telophase, right? So that has to be last. That leaves you with choice C and choice B. Now, does B or C come first? Well, which one do we know comes first? C has to come first. How do we know? Because in B, you have your centrioles on opposite sides, and we know that that occurs after prophase, which we have here. All right? So this is the correct order. Okay, good. So let's now move on to cytokinesis in plant cells. And then we can go back to that question that we couldn't answer before. So cytokinesis in plant cells. So in plants, it's not possible to pinch inward because of the cell wall. Remember back to our organelle unit where we learned about plant cells versus animal cells. One of the biggest distinctions is that plant cells will have a cell wall that surrounds the membrane, right? And the cell wall is very rigid and it is sturdy, and it gives the plant its structure, it gives plants extra protection. So when you think about cytokinesis, how would a cell be able to split into two, a plant cell, if they have that rigid cell wall? How would the cell wall be able to pinch inward if it's so rigid? And the answer is that it doesn't. It can't do that. So in cytokinesis with animals, the membrane, like we saw, creates the cleavage furrow. It just moves inward and it eventually pinches off. Plant cells cannot because of that rigid cell wall. So what they need to do is they need to create something called a cell plate, right? And you can see that right here in the middle here. It's this line, right? So a cell plate will form between the two nuclei. So one nuclei, two nuclei. A cell plate forms, and basically the cell plate acts just like the cleavage furrow. The cell plate will grow outward this way and outward this way, and it will eventually split the two cells in half. So the process of cytokinesis is the same in plants and animals. The only difference is that plants need to form a cell plate within the cell in order to split it, whereas animals can just pinch the membrane inward with a cleavage furrow and eventually split off. All right, so now that we know that, let's go back here. So in the diagram of mitotic cell division shown, which structure is present in diagram B but not in diagram A? And you should easily be able to identify that structure now as this line here between the two nuclei, meaning the answer has to be cell plate, right? Good. All right, so just a couple of more things about mitosis. Mitosis usually takes between 30 minutes and 2 hours, right? So it is not necessarily a quick process. When you think about a lot of things that occur in cells, remember going back to our enzyme unit, we talk about how enzymes are able to speed up reactions and it takes milliseconds, not even. That's a quick reaction. This takes a little more time, right? It's still quick in the scheme of things, right? Replicating an entire cell, copying an entire copy of DNA, organelles, to happen between 30 minutes and 2 hours, still pretty quick, but a little more time consuming, right? Remember, in mitosis, one mother cell will give two daughter cells, always, right? So you have your one mother cell that will go through the process of cell division that we just went through the last 2 days and will eventually give rise to two separate daughter cells that are identical to the mother cell. Very important aspect of mitosis, the whole point of mitosis. Make sure we remember those points. Right? So, the results and the importance of mitosis. So, the results of mitosis is in unicellular plants and animals, it results in new offspring by asexual reproduction. So, if mitosis is occurring in a singular celled plant or animal, then you're going to get this new offspring by asexual reproduction, meaning you will have an exact copy of that unicellular plant or unicellular animal. If mitosis is occurring in a multicellular organism like us, right, it results in the growth and repair of the organism. So you're not necessarily using mitosis in multicellular organisms to get another multicellular organism. We know that that's not possible. But mitosis works in our cells to grow and repair. So if we have cell damage, mitosis will work to repair and regrow those cells that were destroyed. So I'm going to give you an example of that. We are multicellular organisms. If we damage one of our organs in, let's say, a car accident, right, some of our cells will die because of that damage, because of that trauma, right? So mitosis would work in order to repair those cells and grow. So the cells that are alive would go through mitosis and replicate themselves and then create more of those cells. But you're not creating a brand new organism, you're just fixing the cells that you have. So the importance of mitosis. The two cells that are created, the two daughter cells that get created from the mother cell, are exact duplicates of one another. That's one important thing. The other important thing is that this should be ensures, right? So it ensures that the new cells will be able to carry on the same function as the mother cell. That's the other important part. You want to make sure that it carries on the same function. That's why it is being replicated, and that's why the DNA is replicated. It's the exact same cell, and it carries out the exact same function. Okay. How much more are we going to do? Let me see where we're going to stop today. Let's check it out. We're going to stop over here. So let's do 2 more slides. So regulation of the cell cycle. So we just discussed the cell cycle in detail. We went through all parts of the cell cycle. We went through mitosis. We went through cytokinesis. So the frequency of cell division, right, what we just did, it varies with the type of cell you're talking about. Think about skin cells. Skin cells, like we have all over our body, will divide and go through mitosis and the cell cycle all the time, right? Why? What do you think? Because our skin is always being damaged, right? We are always cutting our skin, paper cuts, our skin sheds, we get burns, we get scratches, whatever it is. There's always a need to repair and fix those cells. So skin cells divide all the time. Liver cells maintain the ability to divide, but only do so on rare occasions, right? To repair a wound. Like I said before, if you get into a car accident and you damage your liver, liver cells will start to go through the cell cycle, but only in that instance, right? If you're just sitting normally doing your normal stuff, your liver is not going through the cell cycle. Very specialized cells, such as muscle and nerve cells, do not divide at all. Sorry, guys, I had to pause for a second to cough there. Where were we? The most specialized cells, such as muscle cells and nerve cells, do not divide at all. This is important to understand, specifically nerve cells. I know when we spoke about body systems, we talked about the nervous system a little bit. We spoke about things like Alzheimer's, and we spoke about how that's not reversible, right? dementia, Alzheimer's, these things cannot be reversed because when nerve cells die, they die. And there's no way to get them back. There's no way to repair them. So, this slide here is just to give you a heads up and to let you know that the cell cycle and mitosis is not something that is the same in every cell. It varies on cell, and it varies on length, and it varies on frequency, depending on the cell that we're talking about. Okay? Let's see. Okay. Controls on the cell division. So when an injury occurs, like a cut on the skin, cells divide rapidly. And then when the healing process nears completion, the rate of division slows down. So this is just, again, a repeat of what we were saying before. Cells will divide very quickly in something like skin, right? Cell cycle regulators, right? So what you need to really remember about this is that proteins regulate the cell cycle. So there are many, many proteins found on the inside and outside of cells that regulate the division. Some of the proteins are responsible for starting cell division. Some are responsible for stopping it. Some proteins speed up or slow down. We should know what those proteins are, right? What proteins do we know that speed up or slow down any sort of process, right? Your answer should be enzymes, right? Enzymes are proteins. And the proteins don't send out signals. They keep tissues from disrupting one another. So the whole point of this is that what regulates the cell cycle? Proteins, enzymes. That's what you need to remember, okay? So we are going to stop here for today. Tomorrow we're going to pick back up on, or rather next week, right? We're going to pick back up on this topic. We're going to talk about cancer. We're going to talk about uncontrolled cell growth. We're going to talk about sexual reproduction, asexual reproduction, and then a little bit about how DNA works, right? So please make sure you review this. We are done with mitosis itself. We're done with the actual cell cycle. We are not done with this unit. Not close, but we are done with that. So make sure you review it. As always, please make sure you check Google Classroom. Make sure you are checking Scheduler. Please feel free to leave comments, questions, concerns on the video itself. You can also email us. You could also post on Google Classroom. Whatever you need to do, just make sure you're doing it, right? We are done with our first week of this new remote learning, and I hope that it is working for you as it is for me. We are going to get through this together. We're going to be fine. So make sure you're studying, make sure you're reviewing, and don't hesitate to reach out. All right, guys, have a good weekend. I will see you Monday morning.